Welcome to the deep dive. Today we are really plunging into just a monumental body of work. It's from the Cancer Genome Atlas, TCGA for short. This was huge, a decade-long project, hundreds of researchers looking across 33 different cancer types. The goal, to map out the genomic changes in cancer.
Absolutely. And the core challenge, I think, that this deep dive really gets at, using all the data and tools you've pulled together, is the central question in cancer genomics. How do we reliably find those critical driver genes and mutations, the ones that actually cause the cancer? And how do we tell them apart from all the background noise, the passenger mutations that just kind of happen along for the ride but don't fuel the fire?
Exactly. It's like finding the few key culprits in a massive crowd. So our mission today for you listening is to take this incredible data set, these sophisticated analyses, and, well, give you a shortcut. We want to pull out the most important insights about these cancer drivers, what they are, how they differ between cancers, how they relate to things like the immune system, and ultimately what it could all mean for treatments. Okay, let's dig in.
Yeah, the TCGA effort itself was just immense, coordinating all that data generation, the genomic work across 33 cancer types for, over a decade. It's generated a staggering amount of information and really led to some major findings over the years.
Okay, but with thousands of tumors, millions of potential mutations, how did the researchers behind this specific analysis even start? How would you sift through all that to find the actual drivers? It sounds like a genomic haystack problem.
It absolutely is. And that's where their strategy comes in. They didn't just use one method. They used, I think it was 26 different computational tools. Yeah, 26 tools from groups at 10 different institutions. They broke it down into two phases. First, identifying potential drivers, and then critically validating those predictions.
Yeah.
Using so many tools is really key. It helps handle the big differences in how many mutations different cancers have, and it really cuts down the risk of false positives calling something a driver when it isn't. They even had a specific group, the MC3 working group, harmonize the raw mutation calls from 7 different algorithms before the main analysis, just to make sure everyone was starting clean.
26 tools, wow. Okay, so a multi-pronged attack to boost confidence. But how do you combine findings from that many different tools? How do you decide which genes are, the real deal? Was it just a vote?
It's a bit more sophisticated than just counting votes, yeah. They start with a pretty big list, over 2,100 genes flagged by at least one tool. To narrow that down, they calculated what they called a consensus score for each gene. You can think of it like a weighted agreement score, taking into account how many tools agreed, but also considering the strengths of different tools and trying to spot outliers. And on top of that systematic approach, they also did manual curation. Experts basically reviewed genes that were kind of borderline, comparing them against older TCGA findings to make sure known drivers weren't accidentally missed. So after all that filtering and reviewing, the result was this refined list: 258 consensus driver genes from the automated process, plus another 41 known drivers that were sort of rescued by that expert manual review. So nearly 300 key genes became the focus.
What really jumps out here is how well this multi-tool systematic approach worked. It managed to recapture most of the known driver genes, over 80% in, 20 out of 31 cancer types. That's impressive. But like you said, even this wasn't perfect. It only recovered about 70% of known drivers in stomach cancer, STAD, and in a previous big pan-cancer study. It's a good reminder for you listening, right? Even really sophisticated methods have limits. They can still miss things sometimes.
Absolutely. And conversely, while it might have missed a few, it also found things we didn't know about before. The analysis identified 59 entirely novel potential driver genes never linked to cancer before. Plus, they found known drivers playing a role in new cancer types where we hadn't recognized their importance. This is huge because it could uncover the why for patients whose tumor type might have been missed if we only looked at the usual suspects.
Can you give us some examples? What are some of these novel findings or known drivers popping up in new places? That helps make it concrete.
Sure. For instance, they found ATRX, which is a known driver, seems significant in adrenocortical carcinoma, ACC, that's a rare adrenal gland cancer. They also flagged known drivers like KMT2C, CTMB1, and PTN as important in bladder cancer, BLCA. As for the entirely novel ones, GNA13 popped up at BLCA. Yeah, that's interesting because it's related to GNAQ and GNA11, which are known drivers. RES2 was identified in uterine cancer, UCTC, and it's structurally similar to KRAS and HRAS, huge names in cancer. And KIF1A, a protein involved in transport within the cell, showed up as a novel driver in head and neck cancer, HNSC. It's actually in the same family as another known driver, KIF5B. So you see these connections sometimes.
Okay, so we've got this refined list, some totally new players. Now, is the picture similar across all cancers, or does the type of cancer really change which drivers are most important?
Oh, there's huge variability, really significant. At one extreme, you have kidney chromophobe, KICH, with just 2 driver genes identified. And at the other end, uterine corpus endometrial carcinoma, UCEC, had 55 different driver genes. And what's fascinating is they found a strong link. Cancer types with a higher average mutation burden more mutations overall tended to have more of these consensus driver genes identified.
So more chaos in the genome seems to mean more potential drivers flagged.
Okay.
Are these drivers mostly unique to just one cancer type, or do you see the same ones appearing again and again across different cancers?
It's a mix, really. A lot of them, about 142 out of 258 core genes, seem primarily linked to just a single cancer type. But a pretty substantial number, 87 genes, were found playing driver roles in 2 or more different kinds of cancer. And connecting this to the bigger biological picture, when they grouped the cancer types based on the function of their driver genes, like which biological pathways these genes affect, clear patterns emerged. You saw groups like pansquamous cancers, think head, neck, lung squamous clustering together. Pangynecological cancers, ovarian, uterine, cervical. Pangastrointestinal cancers, colon, stomach, esophagus. This suggests shared biology, right? Similar underlying mechanisms being disrupted by hits in related pathways. And looking at which pathways were hit most often across all the cancers, some really stood out. Pathways involving transcription factors, those master switches for genes, RTK signaling, that's cell communication, often triggering growth, also pathways related to RNA, protein processing, how DNA is packaged, that's chromatin modification, and critically, genome integrity, how cells fix DNA damage. Immune signaling pathways were also frequently hit. So, which are most central? Well, most cancer types had drivers messing with either genome integrity, MAPK signaling, or PI3K signaling. Those are core pathways for cell growth and survival. Squamous cancers, though, they stood out a bit. They had a higher proportion of drivers involved in chromatin modification, receptor tyrosine kinase signaling, and immune signaling compared to other types.
Okay, this is really starting to build a picture. We have the driver genes, how they vary, how they cluster by function. But does the specific mutation within that gene matter? If a gene is a known driver, is any mutation in it bad, or does the location or the exact change make a difference?
Oh, absolutely yes. That's a critical point. Not all mutations are created equal, even in the same driver gene. The precise position, the specific amino acid change it causes, that could be crucial. They really dug into this using another set of tools, 12 of them, focused specifically on predicting the functional impact of individual mutations. These tools used different angles. Some looked at population frequency, some focused specifically on cancer mutations, some even considered the 3D structure of the protein the gene makes. And again, they needed a way to combine these predictions. For the sequence-based tools, they used a combined score, called a CTAT score. For the structure-based tools, they looked at how mutations clustered together in 3D space. So are mutations hitting the same functional spot on the protein? It's like sequence tools read the blueprint line by line, while structure tools look at the finished machine and see where the critical parts are breaking. And they found some really interesting patterns in the types of driver mutations. Missense mutations, those single-letter changes, swapping one amino acid, they actually make up a bigger chunk of the driver mutations in oncogenes, the accelerator genes. Tumor suppressors, the brake genes, they're more often knocked out by truncating mutations, things like frame shifts or stops that basically cut the protein short or garble the message. Although some key tumor suppressors, like the famous TP53 or EP300, PIK3R1, 3-ABP, CASAP8, they also had a lot of important missense driver mutations, so it's not a completely hard rule. And here's a really striking example. The gene CDH1. In breast cancer, BRCA, it's mostly hit by those truncating mutations that wipe out its function. But in stomach cancer, STAD, the same gene is often affected by missense driver mutations in a different part of the protein. This suggests CDH1 might be doing slightly different things, or being disabled in different ways depending on the tissue. It's pretty complex.
That CDH1 example is fascinating. Same gene, different story depending on the cancer type. You mentioned earlier finding drivers where they weren't expected. Did they find specific mutations that looked important, even if the gene wasn't flagged as a top driver for that cancer type overall? Because that seems really key for individual patients, right?
Yes, exactly. And this is a really valuable finding for thinking about personalized medicine. Beyond just the main list of drivers for each cancer type, they found over 1,700 what they call tissue-unmatched likely driver mutations. These were found in over 1,400 different patients. These are mutations where multiple tools predicted, hey, this specific change looks like a driver in this patient's tumor, even though the gene itself wasn't on the main driver list for that whole cancer type. A good example is that RS2 gene we mentioned earlier. It was predicted as a driver in uterine cancer, UCEC, but they also found significant predicted driver mutations in RS2 and several other cancers: uterine carcinosarcoma, UCS, lung squamous, LUSC, lung adeno, LUAD, prostate preade, head and neck, HNSC, testicular tumors, TCGT. In these cancers, RAS2 wasn't previously seen as a general driver for the whole group. It really highlights that if you only look at the known drivers for a specific tissue type, you could miss critical, potentially targetable mutations in individual patients.
That makes total sense. You have to look beyond the average sometimes to see what's driving that specific tumor. Okay, so we have all these predictions, gene level, mutation level. How sure can we be? How do they actually validate these computational findings?
Right. Validation is absolutely essential. You can't just rely on the algorithms. They used an independent data set. This data set contained over 1,000 specific mutations that had actually been tested in the lab in cancer cell lines using systems like Bay3 and MCF10A to see if they experimentally caused cancer-like growth. And the validation results, they were really informative. First, they definitely confirmed the value of the prediction algorithms overall, and they clearly showed that using multiple tools together gives you an advantage. Tools specifically designed for cancer generally did better than tools meant for just any genetic disease variant. Interestingly, the structure-based approaches, looking at the 3D shape, tended to be more specific, meaning if they called something a driver, they were more likely to be right, but they were less sensitive. They might miss some true drivers. It shows the different approaches are complementary. You get a fuller picture using both sequence and structure information. They had some great specific examples, too. Structural clustering flagged mutations in genes like PIK3CA and PIK3R1. These specific mutations hadn't been tested in the lab validation set. But because they clustered physically close to known, validated driver mutations on the protein structure, they become really high priority candidates for future testing. They also pointed out mutations in KEAP1 and NFE2L2. These weren't in the experimental set either, but they had strong supporting evidence from other reputable sources, like the OncoKB database, which classified them as likely oncogenic. That's what we call orthogonal evidence, different lines of proof pointing the same way.
Okay, shifting gears just a bit. The sources also looked at things like hypermutation tumors with tons and tons of mutations and how that connects to the immune system. What did this deep dive uncover there?
Yeah, hypermutator phenotypes are really important. They often have specific causes, maybe smoking history, UV exposure, or, very commonly, defects in DNA repair systems, particularly defects in mismatch repair, which leads to something called microsatellite instability, or MSI. So they specifically looked at MSI status across all the TCGA samples using a tool called MSI sensor. And as you'd expect, they found high MSI levels in the typical MSI cancers: uterine, UCEC, colorectal, code red, and stomach stat. But importantly, they also found significant MSI high rates in some cancers not typically thought of that way, like a fraction of ovarian, OV, and cervical CAC cancers.
And why should you, listening at home, care about MSI? What's the significance there?
Well, because MSI status is strongly linked to treatment response, specifically to immune checkpoint inhibitors, a major type of immunotherapy. MSI-high tumors often respond much better to these drugs. The idea is that all those mutations create abnormal proteins, neoantigens, that make the tumor look more foreign and invisible to the immune system. So they looked at immune markers in the tumor's gene expression levels for things like PD-L1, PD-1, CD8A, which are related to T-cell activity and immune checkpoints, and they found significant differences. In colorectal, stomach, and uterine cancers, the MSI-high samples generally had higher expression of these immune markers compared to non-MSI-high samples. It suggests a more inflamed or immunologically active tumor environment. They also saw some evidence, maybe less strong but suggestive, for increased PD-1 expression in bladder cancers that had a specific mutation pattern linked to ApoBec enzymes, which can sometimes be related to inflammation too. Now, here's where it gets really interesting, I think, and points towards maybe new treatment avenues. They found that hypermutated breast cancer samples, even if they weren't officially MSI high in the classic sense, often did have mutations in genes known to be involved in MSI pathways. And crucially, these hypermutated breast cancers also showed increased expression of immune markers like PD-L1 and CD8A. They saw similar things happening in some cervical and lung squamous cancers too. This hints that there might be subgroups within these cancers, identifiable by high mutation load or specific mutation signatures, that could potentially benefit from immune therapies, even if the cancer type overall isn't usually considered MSI-driven or highly responsive.
That's a really critical insight, isn't it? Finding these genomic clues that might predict therapy response, even where you wouldn't normally expect it. So, Speaking of therapy, did this huge genomic analysis point directly towards specific treatments or drug targets? How clinically relevant is all this?
Yes, that was definitely a major goal to connect the genomic findings to potential clinical actionability. They used established databases, like FILE and Depot, which essentially link specific genetic alterations to known drugs, ongoing clinical trials, or strong preclinical evidence supporting a potential therapy. And the headline finding there was pretty significant. Across the nearly 10,000 TCGA samples they studied, roughly half, maybe 52% based on one database, up to 65% using another, had at least one genetic alteration that was flagged as potentially actionable or biologically relevant. If you narrow it down to clinically actionable mutations with stronger evidence, about 30% of the samples had at least one mutation linked to potential treatment strategies, according to databases like DPO. And what were the common ones? Well, across many cancers, deletions in the CDKN2A gene were frequent, about 13% of samples. PIK3CA mutations, 12%. Amplifications of the MYC oncogene, 8%. BRA mutations or amplifications, also 8%. And KRAS mutations, around 7%. And connecting this to potential treatments. Well, CDKN2A loss, common in brain tumors like GBM, mesothelioma, esophageal cancer, might suggest sensitivity to drugs called CDK46 inhibitors. PIK3CA mutations, really prevalent in uterine, breast, cervical, colorectal cancers, point toward potential benefit from PI3K inhibitors. BRAF mutations, often seen in thyroid cancer and melanoma, are the targets for RAF inhibitors. And KRAS mutations, common in pancreatic, colon, rectum, lung adeno, they're known to predict resistance to certain drugs like anti-EGFR therapy in colorectal cancer, and their role is complex but informative in others. They also highlighted specific examples from the DPO database. Uveal melanoma, that eye cancer, often has GNAQ or GNA11 mutations, suggesting possible sensitivity to MEK inhibitors. Thyroid cancer sometimes has NRAS mutations, which might also respond to MEK inhibitors, maybe even combined with CDK4 drugs. So you can see how this level of analysis starts to bridge the gap from the raw genomic data to potential treatment options.
Okay, so let's try and wrap this up. We've covered a huge amount of ground here. We started with the challenge of finding the key cancer drivers using this arsenal of computational tools. We saw how these drivers vary hugely across different tissues, but also cluster together based on the biological pathways they disrupt. We then drilled down to the importance of specific mutations, not just the gene, and how these tissue unmatched drivers could be key for individual patients. We saw how structural information adds another layer and how all these predictions were validated against experimental data. Then we connected genomic features like hypermutation and MSI to the immune system, potentially identifying new groups who might benefit from immunotherapy. And finally, we saw how this massive genomic map points directly to potentially actionable targets in existing therapies for a really significant fraction of patients.
Yeah, I think the key takeaway is just how powerful this kind of systematic large-scale analysis is. It gives us this incredibly broad view of cancer's genomic foundations. It confirms a lot of what we knew, yes, but it also reveals a ton of novel insights, new potential drivers, known drivers acting in new contexts, new links between mutation patterns in the immune response, and potential therapeutic angles, and it really underscores the sheer prevalence of potentially clinically relevant mutations across so many different cancer types. It strongly reinforces that push towards personalized medicine, basing treatment on the specific genomic profile of that patient's tumor.
So, given that this analysis found roughly half of the tumors already had some kind of genetic alteration with potential clinical relevance, maybe linked to an approved drug, a trial, or even just strong lab evidence, here's a final thought for you to chew on. How quickly and how effectively can we translate these incredibly complex, large-scale genomic insights into practical, actionable strategies for every single patient who walks through the door with a cancer diagnosis? That really feels like the next big challenge, doesn't it?
Indeed.
Thank you for joining us for this deep dive. We'll catch you next time.